In this video, we're going to think about degenerative brain diseases, which often present as dementia. Let's start by thinking, what diseases can cause chronic confusion? There's a list of them here. Many of them you'll have heard of before: Alzheimer's disease, Parkinson's disease, but some of them you won't. The confusion that each of them causes is subtly different. It leads to a slightly different dementia. For example, in Alzheimer's disease, the problem is memory loss, whereas in Pick's disease, there's often an abnormality in frontal lobe processing and emotional control. Parkinson's disease is, of course, associated with movement disorders. So here's the first key concept. Dementias have characteristic presentations and characteristic macroscopic changes in the brain. These pictures show the characteristic changes in Alzheimer's disease. The brain scan shows that as Alzheimer's disease progresses, the brain shrinks and the ventricles increase. And the diagram shows a comparison between a healthy brain and a brain with Alzheimer's. There's overall shrinkage, and in particular, the cortex becomes thinned. The second key concept is that dementias also have characteristic microscopic changes. You can see here on the top the characteristic microscopic change in Parkinson's disease. It's affecting a pigmented neuron, and in the middle there is a Lewy body. The bottom picture shows the characteristic microscopic change in Pick's disease. In that neuron, that kind of swollen look is due to the presence of a Pick body. So what are these bodies? They are abnormal proteins, and this is the next key concept. Abnormal proteins kill neurons. We're going to be coming back to this a lot. So what microscopic changes would provide a definitive diagnosis? There's a list of them here. And when you do the follow-on Kura Cloud lessons, you'll learn more about them. Each of these illustrates the key words that you need to know about this disease. Alzheimer's disease is what we think of as the prototype for dementia. It causes marked atrophy in the brain. But what's really interesting is what's going on at a microscopic and molecular level. It's caused by abnormalities in two kinds of proteins. Abnormalities in the tau protein. That's a kind of structural matrix protein inside the cell. When it's processed poorly, it kind of causes cellular constipation, and the neurons don't like this and stop functioning. And then there's the amyloid-related proteins. So they occur outside the cells. Again, they're processed abnormally and form large, indigestible clumps, senile plaques, and also around vessels as amyloid angiopathy. In Parkinson's disease, it's the basal ganglia, not the cortex, that's affected. And we can see changes macroscopically as illustrated on the right here. The basal ganglia are pigmented. They have pigmented neurons in them. And as Parkinson's disease progresses, this pigmentation is lost. If you compare the two brainstem sections, you can see a normal, darkly pigmented substantia nigra and then a paler substantia nigra in a patient with Parkinson's disease. The protein here that's abnormal is synuclein. So we sometimes call this a synucleinopathy. The abnormal proteins clump together inside the cell and form aggregates called Lewy bodies. Yes, these are the same aggregates that we find in Lewy body disease. It's the distribution of them that means the two diseases have different symptoms. In Parkinson's disease, they accumulate in the substantia nigra, hence the movement disorders. In Lewy body disease, they accumulate mainly in neurons in the frontal lobes, so we get a more obvious dementia. Huntington's disease is another disease caused by abnormal proteins. In this case, it's caused by mutations in the HTT gene, which makes Huntington. The HTT mutation causes a CAG trinucleotide repeat sequence to grow longer and longer. This causes an abnormality in the Huntington protein. It gets bigger and bigger. The elongated protein, when it's then digested by the cell, is cut into smaller toxic fragments which kill neurons in the basal ganglia. You can see in the brains to the right here, the normal one on the top and the Huntington's disease brain on the bottom, that the basal ganglia have shrunken in the Huntington's disease brain. This gives rise to the picture of really large ventricles. We're now going to think about a degenerative brain disease that isn't due to toxic proteins, multiple sclerosis. Multiple sclerosis is an inflammatory disease of the brain. We don't quite know how it's triggered. We do know it has a specific geographic distribution, and we do know that the lesions come and go. In multiple sclerosis, multiple lesions occur throughout the brain and the spinal cord. They occur in the white matter. The white matter becomes inflamed as the inflammatory cells react to myelin, and the brain becomes demyelinated. Multiple sclerosis is a disease that comes and goes. We describe it as remitting and relapsing. This means that the symptoms affecting the optic nerve or the spinal cord affecting urination can come and go. We know that things like heat make it worse, and we know that quite often people get transient weakness. We can see in the brain that the white matter starts looking like the grey matter. Lesions of multiple sclerosis are shown in the brain on the right. Each red arrow indicates a pinky grey patch, which really should be white. We call these lesions plaques. So revising what we've learned today, we've learned that dementias have characteristic presentations, characteristic macroscopic changes in the brain, and characteristic microscopic changes. And we've learned that abnormal proteins kill neurons. We've just done a once-over lightly today. All the research into the molecular mechanisms of dementias mean that we might find treatments one day. Treatments that can dissolve these abnormal proteins or stop them from happening.